Today we're going to be talking about a very important topic in our Principles 1 class and lab, and that's the topic of cellular respiration. Every single organism needs some method for making energy, and that energy is in the form of a molecule that we call ATP, adenosine triphosphate. That molecule can be made by living organisms, depending upon the organism, in a number of different ways. That ATP can be made by aerobic cell respiration, anaerobic respiration, or by fermentation. Larger organisms tend to make their energy using aerobic cell respiration. They have the cellular machinery to do that. They can make much more energy in this manner. And organisms that do this make that energy in a process that we call oxidative phosphorylation, which ultimately allows us to make or net 36 ATP molecules per molecule of glucose that we break down. So what these organisms are trying to do, either by aerobic, anaerobic respiration, or fermentation, is to make that ATP from whatever foodstuffs they are allowed to take into their bodies. Most often and characteristically, we use the molecule glucose to represent respiration. C6H12O6, glucose, can be broken down by those organisms which are capable of doing that in an oxidative manner. And when we say oxidative manner, we mean that they use oxygen in order to do that. When glucose is oxidized by aerobic organisms which have an electron transport system and which use oxygen as the final electron acceptor in that transport system, we're able to break down that glucose molecule into 6 carbon dioxide molecules, 6 water molecules. We get a net gain of 36 ATP molecules, and we always get some heat given off in all these chemical reactions. Organisms that can do this using an electron transport system with oxygen as the final electron acceptor are organisms that can make the largest amounts of ATP, usually a net gain of 36 ATP molecules. Anaerobic organisms use an electron transport system, but they do not use oxygen in that electron transport system. And most of these organisms are small organisms. Most of these organisms are prokaryotes. They can still get enough energy, but they will certainly not get as much energy as they do from an oxidative process when oxygen is the final electron acceptor. Fermentative processes break down glucose usually by what we call substrate level, phosphorylation, and these make some ATP, but only very small amounts, usually only 2 ATPs. The gas given off in this fermentative process is the same gas given off in the oxidative process, but rather than completely breaking down the glucose molecule into CO2s and water, fermentative organisms break it down into some organic molecule, CO2, and small amounts of ATP. Now, there are also some organisms that have a unique ability to do both. There are some organisms that can undergo aerobic respiration, and when there's not enough oxygen available to make ATP through aerobic means, they will shift to fermentative processes. So part of the time they'll be undergoing this process, making maximum amounts of ATP. At other times they'll shift to this when there's not enough oxygen available, and they'll make small amounts of ATP, but this allows them to survive. And organisms that have the capability of doing this are referred to as facultative anaerobes. They can shift processes. Organisms that we'll look at in our Principles 1 lab that have the capability of doing this are the yeast cells. They have the ability, when there's plenty of oxygen available, to undergo aerobic cell respiration, making maximum amounts of ATP. When there's not enough oxygen available, or when they find themselves in a situation where there's not enough oxygen to accomplish this process, they'll undergo fermentation. So they do it both ways, and they can still make enough ATP in order to carry on cellular processes. Organisms have to do this in order to make the ATP. So what do they use that ATP for? Well, the ATP that's made, whether it's made in any one of these three manners, is used to carry on cellular work. It's used to carry on transport work in the cell. It's used to carry on communication mechanisms in the cell. It's used to carry on other chemical processes in the cell. So there are an abundant different reasons why the cell needs energy in order to maintain life processes. And without this process, plants, animals, bacteria would all just be a piece of dead meat. Respiration is a very, very important process. We talk about in class primarily aerobic respiration, and we talk about this also in your lab book. And this aerobic respiration is a little bit more complicated than some of the other processes, the anaerobic processes and the fermentative processes. I want to mention 4 major steps that must occur in aerobic cellular respiration, and they're listed in your lab manual. First of all, the first step in aerobic cell respiration is glycolysis. Glucose is initially broken down. Then, in a second step, which we talked much more extensively about in the lecture class, there is a formation of a molecule called acetyl coenzyme A that must be formed before the third stage of cell respiration takes place, and that third step is the Krebs cycle. This Krebs cycle is a very, very important step. It allows the organism to make reduced molecules like NADH and FADH, and these molecules are coenzyme type molecules that ultimately allow us to make ATP. We can make 3 ATPs for every NADH molecule that's reduced, and we can make 2 ATPs for every FADH molecule that's reduced. What happens in the fourth and final step of aerobic cell respiration is that these reduced molecules are utilized in an electron transport system, electron transport chain, by a process called oxidative phosphorylation. Special enzymes are used to utilize a hydrogen pump that allows us to make abundant amounts of ATP. In aerobic cell respiration, it will be 36 ATPs gained net overall for the process. Without this process, living organisms could not survive. It is very, very vital. The making of energy in all living organisms is required to sustain life.